Hello everyone. First, I wanted to thank you all for taking time out from your busy schedules to listen to this very short webinar. What I want to talk to you about today is figure preparation, and I hope that you'll walk away with some useful tips on how to best assemble your figures for publication. The first thing I wanted to show you is this diagram of the research life cycle. Now, I'm not going to walk you through every step of the process here, but I wanted to show you this diagram just to press upon you how much science really builds upon itself. Because of this constant building, it's really important to make sure that your data are rigorous and presented transparently so that others can expand and develop upon your work. One way you can do that is by preparing a really nice figure for your paper that clearly represents the actual experimental results you obtained. One of the take-home messages I hope you get from this webinar is that it's going to be pretty near impossible to make a nice figure unless you've gone through all the right steps in data collection. So about a year ago, JBC started screening every image in our article slated for acceptance. We're not just looking for image manipulation, we're also looking at image quality, making sure that authors are adhering to our policies, and just generally, will this figure reproduce well when it's published? We really want your data to look the best they can by making sure there are no artifacts in your images and that they're the proper size and resolution. Essentially, our workflow is that once an associate editor makes the decision to accept a paper, it's screened by one of our analysts, and if everything checks out okay, the paper is formally accepted and published online as a paper in press. Now, a small percentage of our papers are declined at this point for inappropriate image manipulation. So far, I'd say it's about 4% of our conditionally accepted papers. Now, I'd like to say that the other 96% sail through image analysis and are accepted immediately. The reality is, we send back most of our papers for some sort of correction. Sometimes it's not for adhering to our editorial policies, but the vast majority is for problems with image quality. Here's a list of the common image quality problems we see, and they fall basically into two categories: not properly acquiring and/or saving the image, or not properly preparing the figure. I'm going to harp on this point a lot, but the most important thing I want to point out about figure preparation is that it begins when you acquire your data. In fact, most of this webinar is going to focus on data acquisition, because if your image hasn't been properly acquired, saved, or managed, then it'll be impossible for you to make a publication quality figure in the end. So I really cannot stress how crucial this step is. So here are some questions you should be asking yourself as you're acquiring your data. Is it at the correct resolution? Are you in the linear or dynamic range? Is anything over or under saturated? And are you saving your files in the correct format? Before I jump into these points, I just wanted to mention two things that are important to understanding what makes a good and bad image. First things first, an image is comprised of pixels. So if you zoom here, you will see this grid of pixels or boxes that when you put it all together will show you that your co-IP experiment worked or your co-localization experiment worked. Each of these pixels contains information that tells the computer what intensity or color of gray to display. So that's number one. The second thing you'll need to understand are histograms. The histogram is a distribution graph that tells you how many pixels in your image are completely black, how many are completely white, and then every shade of gray in between. Here's a histogram of an image that's been appropriately acquired. Nothing in it is completely black or white, and you can see that the pixels aren't concentrated at either end. Now, if the histogram had been shifted in either direction, it means that your image is either over or underexposed. So first, when you're acquiring your image, you want to start off with the right exposure. The histogram will tell you if the image is in the right range. Here is an example of two different exposures of the same blot developed using film and scanned using a flatbed scanner. For the one on the left, from the histogram you can tell that none of the pixels are completely black or white. The histogram also shows you that the pixels aren't gathered at either end. As for the one on the right, the spike at the end where all the black pixels are tells you you have an overexposed blot. Why is it so important to ensure you have the right exposure? First, if you look at this pair of images, which one is more informative? Now these are cells that were stained with DAPI. If you look at the one on the left, it shows a lot of the fine detail of the nucleus of the cell on the bottom, and you can actually see the chromosomes condensing in the cell on the top. Whereas if you look at the one on the right, all that nice detail is missing. Second, if at any point you want to quantitate your data, you will be underestimating your signal if your image is saturated. The reason is that once your film or detector has collected its maximum value, it's impossible for it to detect anymore. So this is kind of a ridiculous case, but let's take a look at this overexposed blot, for example. The histogram here tells you that the most abundant pixel intensity in this image is 0 or pure black. And if you then attempt to perform densitometry on this upper band, something I would definitely not recommend, and then normalize all the values to the first band, there's hardly any variation in intensity. Now if you then take a more appropriately acquired image and do the same calculation, there's now variation in the amount of protein being detected. Remember, there's a threshold as to how much signal your film or detector can collect. Once you've surpassed that threshold, you will underestimate your signal and you won't be able to tell by how much. If you're using an imaging system, it's usually a bit easier to tell if you're in the right range because the system usually will display the histogram next to the image as you're acquiring it. For example, here's an image that was acquired in Lycor. Now for Lycor, the histogram next to the image shows you all the information that's contained in the blot, and the line is what tells the system how much of that information to display. For the top image, pretty much all of the information that's contained in the blot is being displayed. As you increase the contrast in the system, the image no longer displays all these data points down here on the left of the histogram. In fact, if you look at the image, one of the bands even disappears. This is not a good way to display your data because now you're cherry-picking your data. Remember, it's critical to ensure that you're showing the actual experimental results in your figures. Dirt, specs, background information are all hallmarks of true data. The next thing you want to do is make sure you're acquiring your image at the correct resolution. Resolution is the density of pixels in your image. For example, 10 dpi would be 10 pixels per inch. Keep in mind that most journals will require that you submit your figures with a minimum resolution of 300 dpi. That means you need to be acquiring your data at a minimum of 300 dpi. Please be aware that some imaging systems ask you to specify upfront at what resolution to acquire your data. For example, the Typhoon will ask you to specify the size of the pixel before scanning. Be sure with these types of systems you're acquiring the image at a minimum of 300 dpi since you won't be able to go back later and rescan the image if it's not of sufficient resolution. In the case of the Typhoon, that would mean scanning with a pixel size of 50 micrometers or less. You may have to wait a bit longer for the scan, but it sure beats having to repeat the experiment because the resolution isn't good enough for publication. If you're using an imaging system, one other topic we should cover is binning. Binning is a process performed by the system that speeds up acquisition time as well as increases camera sensitivity, but it does this by sacrificing spatial resolution. Here's a simplified example of how it works. Let's take this image and let's say that each box here represents a pixel. If you bin the image one by one, that means the camera will acquire the data in 1 pixel increments, and you end up with an image that resembles the original. If you bin the image 2 by 2, what the camera does is combine a 2 by 2 pixel block and make it into a superpixel. This will speed up the acquisition process because now it has less blocks to scan, and because it's combining all the pixel values together, it will increase its sensitivity. But for my example here, by binning 2 by 2, you've decreased the resolution of the image. Here are some examples of what binned images look like. These plots were acquired on an image quant in super mode. The final resolution isn't great at 217 dpi, but because it was binned 4 by 4, meaning a 4 by 4 pixel block was combined to make one big pixel, the bands ended up looking really blocky. If you're using an imaging system, remember to check the binning before you acquire your images, and make sure it's set 1 by 1. You don't want to end up with a blocking-looking image at the end. As a reader, these images aren't very pretty, but from an image analysis standpoint, these look unnatural because the bands look rectangular and the background is flat. In fact, we often have to clarify these types of images with authors by requesting the original data. Okay, so now you've got all this cool data. Are you saving it in a way that's going to be usable in the future? First, you should always save your file in a universal file format that uses a lossless compression such as TIFF. That means avoid JPEG and PDF at all costs. If you're using an imaging system, you should also save a copy in the native format in case you need to go back to later. There are a few reasons why you should avoid using JPEG and PDF. One, the algorithm used in JPEG compression fundamentally changes your data. Take, for example, the image of the word JPEG. Here it's been saved as a TIFF. When you look at the histogram, it shows you that the image is made up mainly of black and white pixels and a few intermediary shades of gray. When that image is now saved as different quality JPEGs, the histogram shows you that pixels are being introduced that never existed in the original image. So how is this going to affect your scientific data? So here's the same image I've saved either as a TIFF or various quality JPEGs. I then did a surface plot analysis of the blots in image J. You can see even in the surface plot for the high quality JPEG saved, the data already looks different than the original capture. And then when you save it at progressively lower qualities, the blot starts looking blocky and pixelated. We would definitely flag these images during screening. Not only does one save in JPEG alter data, but each time you save as a JPEG, that compression is getting reapplied. Now here's a set of images that I saved 10, 50, or 100 times as a JPEG, and if you blow it up, you can see that with each save, the pixels are changing. Now I don't think you're going to be sitting at your computer and hitting Ctrl+S 100 times, but think about how many times a file gets opened and saved. Now each and every time you're saving that file as a JPEG, the computer is reapplying that algorithm and you're going to end up with an image that no longer resembles the original capture. Now I also mentioned not saving as PDF files either. The reason PDF should be avoided is because the settings aren't always transparent. Here's just a portion of the settings found in Adobe Acrobat. But these settings may be harder to access in whatever program you're using to write the PDF file. Also, some of these settings can't be changed in certain programs. Some of them will automatically apply JPEG compression to the images or downsample them to less than 300 dpi, amongst a million other things. So just to avoid any possibility of compromising your data, I would always save as a TIFF. Otherwise, you're going to have to sift through all these settings to figure out what's causing an artifact to appear in your image file, and it may not be correctable. So your ideal image file should have a minimum resolution of 300 dpi. It should be in TIFF format. Remember to save it as RGB or grayscale, not as index color. Finally, you want your image to be binned one by one to avoid looking like a Lego set. Think about your own imaging systems and set these as goals that you want as your final output. Since you're likely using shared equipment, I would also double check these settings before you start acquiring your images each and every time. The last point I'd like to touch upon in image acquisition is data management. First, you want to make sure your data are secure. Remember to back it up. You'd be surprised how many authors can't produce original data for papers that have not even been accepted. Also remember, 5th year PhD-U will thank 2nd year PhD-U if you detail your experiments properly, label your data, and archive it appropriately. Not only will future you thank you, but postdoc number 10, who joins the lab 2 years after you leave and inherits your project, will also thank you. So that was a lot of stuff I threw at you, but these are the main points I want to reiterate. Make sure you are acquiring your image at the correct resolution and exposure. Keep in mind what makes up an ideal image file. Make sure you have a data management plan in place in your lab. Funders and institutions may have their own mandates about how data should be managed. Also, many journals have data availability requirements for the papers they publish. For example, the JBC requires that all authors maintain all of their original data for a minimum of 6 years after the final publication date of their article. So now that you are all experts and have the basics down about image acquisition, I wanted to talk to you about figure preparation. So these are the big pitfalls that we're seeing when we're screening images. Remember to always read the instructions for authors for the journal to which you would like to submit. These will tell you their policies on how you should be preparing your figures. For example, at the JBC, authors should declare all gel splices, and splicing should only be used when it's showing the same gel or blot. Other problems we often see are authors fiddling with their images too aggressively, issues with image resolution, and the use of graphics programs not intended for handling high-resolution images. So here's a set of the same images, just adjusted different ways. If you were reading a paper, which image would you prefer to see? So I'm hoping that the majority of you would prefer to see the one on the top left. This is the original scan of the film and hasn't been adjusted in any way, while the other three were all heavily adjusted. Remember, the final image you present your paper should look as close as possible to the original data, warts and all. The more you adjust your image, the more chances you also have of introducing artifacts that may be flagged later. Another thing you always have to keep in mind is the resolution of your image during figure assembly. Remember, the better the resolution, the crisper your results will look in your paper. Let's look at this example. Say in the original draft of your manuscript you had a 4-panel figure. Once you get the reviewer comments, you realize the figure actually works better as only a 2-part panel. So you decide to increase the size of the blots you had in A by 2. Now if you just go into your figure file and resize the image, what do you think happens to the resolution? The resolution is actually halved, so your 300 dpi image is now 150 dpi. So if you want to increase the size of your images, you'll have to rescan or re-export your blots at a higher resolution. So keep the resolution of your images you have in your figure in mind as you're assembling them. One common misconception I see with authors is that they think they can just go into Photoshop and just change the resolution by entering whatever number they need in the resolution box. This is absolutely not true. Take for example this picture of my dog when he was a young puppy. The image is currently at 72 dpi, typical of figures prepared in PowerPoint. Let's say you're about to submit your figures and you realize, oh crap, my figure's the wrong resolution. No problem, I'll just go into Photoshop and resize. Now there are actually 2 options. If you have this resample box unchecked, Photoshop will not create any new pixels, but will instead resize your image to the resolution you enter. This is the best option, but what will happen is that you may end up with a figure that's too small for publication. On the other hand, if you check this box, Photoshop will keep the physical dimensions of your figure the same and will artificially increase the resolution by creating pixels. This doesn't actually make your figure 300 dpi because if you look at this image closely, it's still a bit blurry. Also, you will run the risk of introducing artifacts into your figure that may end up being flagged by editorial staff. Since we're talking about software, make sure you're using a program that is intended to combine vector graphics like text and line art with raster graphics like images. This means avoid using PowerPoint for anything except for making live presentations. The reason why you should avoid PowerPoint is because it automatically saves any image as 72 dpi, which is unsuitable for publication. In fact, your figures and labels will end up looking really blurry. Another thing I often see from figure files made in PowerPoint is aliasing artifacts. So sometimes PowerPoint just can't handle small rotations of an image, and it'll create these small stair steps. I'll even do this with text and graphs. So you won't run into this issue if you're using appropriate software. So remember, PowerPoint should not be your go-to when you're assembling your figures. If you don't have access to programs like Illustrator or Canvas, no worries. There's free software out there to download. Inkscape is an Illustrator equivalent, and GIMP is a Photoshop one. One final point I like to bring up is the type of file you're submitting to the journal. Although the JBC does allow authors to submit figure files as PDFs, as I've already mentioned, there are many different settings and PDF conversion engines that may introduce artifacts into your figures. I've seen PDFs that make it look like there are gel splices, lowered the resolution of the image, introduced JPEG artifacts, and even turned saturated pixels of an image into editable text boxes. So I would strongly urge you to submit your figures as TIFF or EPS files. Are you a reviewer evaluating a manuscript for a journal? Here are some suggestions for when you're reviewing the paper. Go through the manuscript just once, just looking at the images and not reading the text. This will allow you a chance to concentrate on just the images. If you divide your attention between the text and images, you might miss something. You'd be surprised how many figures get to image screening with a duplicated image right next to each other that pass 2 or maybe 3 reviewers. Be wary of overly adjusted images or overly cropped blots. Makes you wonder if the authors are trying to hide something. And it's not just blots, still images can also be manipulated. Finally, blow up the figures on the screen. It'll be a lot easier to see figure irregularities. Also, don't be shy to ask the authors to revise the figure if you don't think it's up to snuff. If you think anything's been cropped too closely or the background is just completely wide on a blot, I would definitely recommend including those comments in your review. So just to wrap up on figure preparation, less is more when it comes to images. Adjusting your image more doesn't make it look better. Keep track of the resolution of your image once it's been inserted into your figure. Use the appropriate software and submit your files as TIFF or EPS. Finally, once you're done assembling your figure, take a look at it. Does it resemble the original capture of your data? If not, I'd go back and restart. And I'd like to close with this final thought. Remember, garbage in, garbage out. You will not be able to make a good quality figure if your starting materials aren't good. That means good resolution, proper file format, and appropriately adjusted images. So thank you all for your attention. If you'd like more information on the topics covered today, please stop by these two websites.